The second important tool in rDNA technology is vectors. So vectors are cloning vehicles. They are DNA molecules. These vectors are called cloning vehicles because they carry the targeted DNA or a foreign DNA which has to be cloned. So they help in amplification of the inserted DNA segment because these vectors have self-replicating property. Coming to the ideal features of an ideal vector, it should be a low molecular weight DNA and it should have unique cleavage site for the activity of restrictions endonuclease. They should have self-replicating property when it is introduced into the host cell, and also they should possess antibiotic resistant gene. So this antibiotic resistant gene helps in selection of transformed cells from untransformed cells. But very rarely we can have all these in one vector, but we can always modify the vector to have all these properties. Coming to the various vectors we have or which is utilized in rDNA technology, they are plasmids. Second one we have bacteriophages. The third is cosmids. Then we have artificial chromosome vectors. So all these can be expressed. When they are expressed, we call them as expression vectors. Now we will learn one by one the details of these vectors. Plasmids. So commonly we have bacterial plasmids. They are extra chromosomal, small, circular and duplex DNA molecules. There are several properties which make them extremely useful as cloning vectors. For example, they have origin of replication and they confirm antibiotic resistance gene. They can be existing as single or multiple copies. So sometimes they can be 10 to 20 copies per cell, and they have a special feature that is self-replicating. So these plasmids can replicate independently from the bacterial chromosome or bacterial DNA. So apart from naturally occurring vectors, we can also artificially restructure them, modify them, and we have few modified plasmids. For example, PBR 322, PUC 19, PUC 101. So these are the commonly used artificially structured plasmids. Coming to the nomenclature of plasmids, we will see how these plasmids are named. So we will take an example of a E. coli plasmid, that is PBR 322. So this small p which is written in lowercase, actually it is denotes the plasmid. So plasmid is designated as P in a lowercase. Now the BR which is written in capital letter, they denote the first letter of the discoverer. So it's a first alphabet of researcher's name, Bolivar and Rodriguez. Now we have a number, so that is the number given by the researcher. Sometimes some plasmids are given names of places where they are discovered. For example, we have PUC 19. So UC is actually a place where it was discovered, that is University of California. So it can be a name of the researcher or it can be a name of the place where it was discovered. PBR 322 is the most commonly used plasmid as a vector in recombinant DNA technology. Why so? Because there are so many features, like it is very small size, that is it is having only 4361 base pairs. So if it is very small, it helps in easy incorporation into the host cell. It has many antibiotic resistant genes. For example, it has ampicillin resistant gene, it has tetracycline and chloramphenicol resistant genes also. So these antibiotic resistant genes can be used as a markers when we want to identify the host, whether it is transformed or not. What I mean to say is, if you want to pick up an intact plasmid or a recombinant version of the plasmid in a host, these resistant genes will be very, very helpful. Apart from this PBR 322 has many recognition sites for various restriction endonucleases. For example, it has recognition sites for EcoR1, HIND, HIND3, BAMH1, PST2. So all these features make this E. coli plasmid BR322 a most commonly used plasmid in recombinant technology. Another vector of choice whenever we are dealing with large DNA segment is bacteriophage or lambda phage. So this is one advantage over plasmid, where plasmids cannot accept or carry a DNA segment more than 10 kilo base pairs. So these lambda phages, they are the viruses that replicate within the bacteria. And if you see the genome material of these lambda phages, they comprise of linear and double-stranded DNA. And if you see the base pairs, it is somewhere around 48,513 base pairs. And one more features which make this as a choice is the genome pattern. So if you see the lambda phage genome, 1/3 of this genome is not essential. So when we have such a feature, we can remove that and we can add the foreign DNA at that place. So this feature of bacteriophage makes it a choice for our choice as a vector in rDNA technology. So we can remove the non-essential DNA and we can insert the foreign DNA without affecting the properties of the phage genome. So the DNA fragments which are present at these ends, they are called as Cosmids, which actually form the packaging signals. So they are very essential for the packaging of this DNA into the phage particle. So we should retain these ends, and the in between, whatever it is in a non-essential, can be removed by a restriction enzyme, and that cut end can be joined by a foreign DNA. Now this recombinant DNA or a hybrid DNA or a chimeric DNA can be packed into the lambda phage. Another type of vector is Cosmids. Cosmids, they are the artificially structured circular DNA. So it's a combination of plasmids and bacteriophage. So they will take the useful features of both plasmids and bacteriophage lambda. So they will have origin of replication and plasmid with cos sites. So cos sites are present in bacteriophages, which are required for packing this DNA into the phage particles. Apart from this, they will also have antibiotic resistant gene, which can be used as a marker to identify the clones. And there are several restriction sites where foreign DNA can be inserted. Apart from this, Cosmids can carry up to a desired DNA or foreign DNA of 30 to 50 kilo base pairs.